let's go ahead and jump in. All right, so here we are, week 8 of this term 3 of the Excel Center. And our final notes, the final thing that we're going to cover is sort of the basics of DNA. So we've talked about DNA a little bit in this class before. We talked about it when we went over the macromolecules. DNA is a type of nucleic acid, which is one of those 4 things that we talked about. We talked about it when we went over the parts of the cell. So we mentioned that the DNA is always inside of the nucleus in eukaryotic cells, and that it's kind of floating in the middle of prokaryotic cells. And we've talked about that DNA can be found in all cell types. Every single cell on planet Earth has DNA in it. But today I want to give a little bit of a brief overview of what exactly is DNA, this little spiraling structure that we've heard so much about so far. So the DNA, the structure of DNA, what it looked like was a mystery to scientists for many, many years. They knew it was there, they knew it was inside of cells, but they weren't quite sure exactly what DNA looked like. It wasn't until the 1950s that a scientist named Rosalind Franklin used a special type of photography and visualization called X-ray diffraction. And she got this famous picture of DNA showing how it kind of twists around itself. So she got this sort of X-shaped picture, which led to the idea that there's 2 strands that kind of cross over with each other. So based on her picture, other scientists named Watson, John Watson and Francis Crick, were able to build this sort of 3D structure of DNA. So they were able to figure out that there's these 2 strands here, and they kind of circle and cross around each other. It looks sort of like, if you've ever seen like a tall spiral staircase in a movie or a TV show, that is sort of what DNA looks like. So it's these 2 different sides that kind of wrap around and cross over each other. So DNA has a big long name. It is called, the DNA stands for deoxyribonucleic acid. Obviously, that's a big mouthful, so scientists ever just really call it DNA. The N and the A are where the nucleic acid part comes in. So remember when we talked about the macromolecules, nucleic acids were one of those macromolecules. And so the NA in DNA stands for nucleic acid. And deoxyribo stands for a type of sugar that DNA is made out of. So the reason why we have DNA in our cells is to store and carry information in our genes. So genes are segments of DNA that have the instructions to make a certain protein. And if you remember when we talked about the structure of the cells, those ribosomes, those tiny little ribosomes in the cell, those are what actually make the protein, but they get their instructions from DNA. So all of the thousands of different proteins that your body makes in order to turn you into a human live being, all of that information is stored in the millions and millions and millions of bases that make up DNA structure. So this twisty, curvy helix of DNA contains all the information needed to make you. So DNA is a nucleic acid. And you may remember that nucleic acids are a macromolecule, which means that they have monomers, little individual base units, that get built into long chains called polymers. So monomers, our basic unit, are called nucleotides. Nucleotides have three parts that we'll talk about in a second. They've got a phosphate group on one side, a sugar in the middle, and a nitrogen base on the other side. Now, if you take thousands or millions of these nucleotides and put them together in a big long chain, you get DNA. Or in some cases, DNA's single-stranded cousin, RNA. So DNA and RNA are both nucleic acids. That's what the Na stands for. DNA has two strands. RNA just has the single strand. But each one of these little lines inside the DNA, each one of these little colored lines, represents a single nucleotide. So we have our nucleotides, put thousands and millions of them together, and you get nucleic acids, either DNA or RNA. So a nucleotide has three distinct parts to it, like I mentioned a second ago. We've got our phosphate group on one end. We've got our sugar, sometimes called a 5-carbon sugar, or sometimes called a pentose sugar, because as you can see, there's 1, 2, 3, 4, 5 different sides on this particular sugar. And then the third part is called a nitrogenous base. It's a type of structure that's based on nitrogen. And based on the exact structure, there are 4 different types of nitrogenous bases, and they're going to be really important as we talk about them today. They have longer names that we'll talk about in a second, but you'll usually see them represented by A, T, G, or C. So every single nucleotide has the exact same phosphate and sugar setup. A lot of times these are called the sugar backbone or the phosphate backbone. So these ribbons, these blue ribbons right here on the DNA, represent the kind of phosphate and sugar together. And those are the exact same for every single nucleotide in DNA. The only thing that is different is this base over here on the side. So you can have one of 4 different bases: an A, T, a C, or a G. So again, you have these nucleotides that are made out of 3 different parts. And if you connect millions of these nucleotides together across from each other in these double strands, you get DNA. Anybody have any questions about DNA or nucleotides so far?
I'm good.
No, sir.
Okay. So the sugar in DNA and RNA is this 5-sided sugar, looks something like this. The only difference between them, and the reason why DNA has that D, is that in RNA, the R stands for ribose sugar. That's the type of sugar that this is. You'll notice it has this little OH group down here. In DNA, the O in that OH group is missing. It's just an H. So instead of being ribose, this is ribose minus an oxygen. So scientists called it deoxyribose. basically just a fancy way of saying ribose that is missing an oxygen at this part right here. So the D in DNA comes from deoxyribose, meaning the sugar missing this particular oxygen down here. So, like I mentioned, we have 4 different types of bases in DNA. We have adenine, which is our A, guanine, which is our G, cytosine, our C, and thymine, which is our T. So some of these are made of these like 2-ring structures. Some of these are made of 1-ring structure. You don't have to know which one is which. Just know A, G, C, and T are what make up the bases. And so when you look at a strand of DNA, like I mentioned, these phosphates and these sugars are connected to each other, and they're kind of what's going down the sides of the DNA. These bases are what are crossing over in the middle. So all the A, G, C's, and T's are in the middle of the DNA. And then all of our phosphates and sugars are kind of going around the outside part of the DNA. So if you uncurled the DNA, made it straight, like ironed it out, it would look something like this. We have sugars and phosphates connected to each other, running up and down the sides of the DNA. We have these bases, which are connected across the middle of the DNA. And since there's 2 strands, there's 2 bases. So a lot of times they're called base pairs. And then remember that each one of these phosphate sugar base groups is called a nucleotide. So this is a nucleotide, this is a nucleotide, this is a nucleotide, and so on and so forth. So our base pairs line up across from each other in the middle. So does anybody notice, feel free to answer either out loud or in the chat, does anybody notice a particular way in that the bases are pairing up with each other, or the types of bases that are pairing up with each other?
Yeah, when it's like A, it's like T. When it's G, C, T, A. So they're just like going like this way.
So you mentioned that the A's and the T's look like they're bonded up together. What about the C's and the G's? Are they doing the same thing?
Leave her alone.
Yes.
Yeah. So as those guys just pointed out, in DNA, you'll notice in this picture, the A and the T are always together, either on one side or the other, doesn't matter. But wherever there's a T, there's an A, and vice versa. Same thing with the G's and the C's. So the G's and the C's are together in this picture as well. So that is an extremely, extremely important part of DNA, and it's called the base pair rule.
Mr. Michael, can you go back for like a second, please? On the hydrogen bonds, or what it's called, like it's look like on the G and C, like it's like more than the A and T.
That is true. So I'll talk about hydrogen bonds in a second.
Oh, okay.
Basically, the forces that hold the nucleotides together. And it is true that the way that they're structured, G and C have 3 bonds between them, and A and T have 2. It doesn't really make a super huge difference to the DNA overall. That's just sort of how things lined up when these bases came together. So like I mentioned, we will talk about hydrogen bonds in a second here, but the important thing to note is that A and T go together and C and G go together. Like I said, this is called the base pair rule. It means that A and T T and A always, always, always go together in DNA. It should never, ever, ever be different than that. T and A always are going together. Same with G and C. G and C will always line up across DNA to each other. If they ever don't line up this way, that means something's wrong with the DNA. The DNA is a little bit broken in some way. But in normal, healthy DNA, you should always have A's and T's together and G's and C's together. So that's sort of what our little picture over here is showing. You have on the one side, on this first one, you have A, and then T is on the other side. And you can see by the shape that only the A's and the T's fit together. So the T is a little point, the A is a little indent, so they have to go together. Same thing with the C's and the G's. So in this little picture, the C is this little rounded, and the G has this little round cut out of it. And so the G and the C line up perfectly together. So always, always, always in DNA, you'll know that if there's a T on one side, there has to be an A on the other side. If there's an A on one side, there has to be a T on the other side. If there is a G on one side, there has to be a C on the other side. And if there is a C on one side, there has to be a G on the other side. Always, always, always in DNA, the bases will line up this way. All right, so like Nasima pointed out earlier, the bases are held together in the middle of DNA by structures called hydrogen bonds. Oh, I almost forgot. Okay, so I'm going to have you guys practice a little bit with me with our base pairs. So we're going to take these sets of base pairs, 3 at the time, and I'm showing you what one side of the DNA looks like. And then I want one of you guys to tell me what the other side of the DNA is going to look like. So if one side of our DNA goes A, T, A, what is the other side of our DNA going to look like?
Was it TAT?
TAT. Bingo. A goes with T goes with A goes with T. All right, our second group, we have TCA. So what would the other side of our DNA look like?
A, G, and T.
Yeah.
Perfect. A, G, T. T means you have an A. C goes with G. A goes with T. All right, third set over here, we have T, G, C. So what's going to line up on the other side?
ACG.
Bingo. A, C, G on our other side. T goes with A, G goes with C, C goes with G. And then finally, our last group over here, we have G, G, C on one side. So what is going to be on the other side?
C, C, and G.
Perfect. C, C, G. G goes with C, G goes with C, and C goes with G. So T and A and C and G always, always, always will line up that way on the DNA. So does anybody have any more questions about the nucleotides or the base pairings? Seems like you guys have it down pretty well, but does anybody have any questions on how these bases line up?
Not for me, thank you.
Alrighty. All right, so like Nasima mentioned earlier, hydrogen bonds are what keep the bases on the DNA together. These are what link the ATs and the GCs. So hydrogen bonds are a type of chemical bond that happens when you have a hydrogen to one element, so it's bonded to one element, but there is another element nearby that it's kind of attracted to. So like in this case, we have, I think this is a G and a C. So these nitrogen groups and oxygen groups over here and over here have hydrogens on them that are kind of attracted to the other element. So normally, in most cases, hydrogen bonds are very weak. They're a very weak form of chemical bond. It's very easy to break them apart. But in DNA, there are millions of them going all along that strand of DNA. So because of that, they're able to hold the two strands of DNA together really, really well. So it means that the cell has to make special proteins in order to break the DNA apart if it ever has to get in there and read some of it. And so the hydrogen bonds come from hydrogen atoms being attracted to nearby atoms. It's sort of like when you hold two kind of like small magnets close to each other, you can feel that they're a little bit attracted to each other, but it's still easy to pull them apart, right? So these guys aren't directly bonded to each other, but they have these kind of weak attraction forces due to the hydrogen bonds. So again, hydrogen bonds are very weak. CG has 3 of them. AT has 2 of them. That's not really super important. That just kind of happens due to how their structures are set up. But they're able to hold the DNA molecule together just by having so many of them. All right, so we're not going to get super detailed into how exactly the DNA code works in this class. If you take Biology B, in a future term, we'll go really in depth into how exactly the DNA turns this code into proteins. But essentially, it all comes down to different combinations of these ATGCs. So the genes in your DNA are segments that have a long stretch of DNA that's the code for a protein. And sometimes, depending on how exactly that DNA is set up, you can code for different types of proteins. So for example, if you have like this weird alien type creature that has DNA in it, then maybe some aliens, maybe their hair color gene goes CAT. And if you have that gene in your DNA in your hair color section, you're going to end up with purple hair, like this guy does down here. However, maybe other creatures have a different DNA code in their hair gene segment. Maybe their code goes TAC. And if you have that code, then you have yellow hair. So these differences in exactly how the bases are aligned lead to the differences between people, between species, between families and groups and organisms, all to differences in how the DNA is structured. Again, every single animal, plant, microorganism on Earth has DNA and uses DNA for their proteins. So the differences in them come from the different ways that the DNA is arranged and is read. So it might seem crazy that these 4 different bases, A, T, G, C, determine everything about how all organisms on Earth work, but I kind of like to think about it this way. So these bases, we assign them these letters. And in English or Spanish or whatever language that you have, you use letters to form words. And then those words can form sentences. So even though, say, in the English language, we have 26 letters in it, but by rearranging those 26 letters in different ways, you can get millions, or I should say thousands of different words and millions or billions of different sentences, right? All the books that have ever been written are different because the authors put the words in different orders and changed up how the sentences work. So just based on the 26 letters in English, we can get books that are thousands of pages long, filled with all sorts of different words. The same concept kind of works for our DNA bases. A single gene might be hundreds or thousands of bases long. And so by changing up where your T's go versus where your G's go versus where your A's and C's go, just by changing up the order that they're in, you can create thousands of different proteins that are responsible for all sorts of different jobs and different responsibilities in your cells. So the information that goes to the ribosome to make those proteins, it all comes from different combinations of your base pairs inside the genes, inside your DNA. All right, does anybody have any questions about DNA or base pairs or kind of the basics of the genetic code so far? All righty. So if not, that pretty much brings us to the end of our notes today. Early one, I know, but I figured I would give you guys a little bit more time to look at the notes and work on the final. And I didn't want to introduce too much new information this late in the term. So the assignments that you have for the rest of this week are pretty simple. I wanted to keep them short and easy so that you had as much time as possible to work on the final. Your assignments for tomorrow and Thursday are very simple little questions about kind of the course in general. What are things that you liked or didn't like? What's something that you felt worked really well in terms of how I taught the course? Stuff kind of like that. And then your assignment for today is another pretty simple one on DNA base pairing. So much like those examples that we just did in the notes, I give you a string of DNA strands, and then I want you to write in what would be the complementary strand. So what strand would match with this one that I've given you? And that's it. That's all you need to do for the homework for today. All righty, where are we at here? There we go. So does anybody have any